Welcome to our podcast on drug discovery innovations. Today, we're discussing how microdroplets technology is revolutionizing the search for new drugs through high-throughput screening on targets of interest. I'm joined by Dr. Alexandra Kim, a leading researcher in microfluidic applications for drug discovery. Thank you for joining us, Dr. Kim.
Thank you for having me. I'm excited to share insights on how microdroplets are transforming the drug discovery landscape.
Let's begin with the basics. For our listeners who may be new to the topic, can you explain what microdroplets technology is and how it differs from traditional screening methods in drug discovery?
Certainly. Microdroplets technology involves the generation and manipulation of tiny droplets, typically in the picoliter to nanoliter range within microfluidic systems. Unlike conventional bulk assays, where reactions occur in milliliter scale volumes, microdroplets allow us to compartmentalize reactions in thousands or even millions of individual droplets. Each droplet essentially acts as an independent microreactor. This miniaturization not only dramatically reduces the amount of reagents and samples needed, but also enables high-throughput screening with rapid reaction times and precise control over reaction conditions. In drug discovery, this means we can test a vast array of compounds against a target of interest, much faster and with greater efficiency than traditional methods.
That's fascinating. So instead of performing thousands of tests in large volumes, you're effectively running millions of mini experiments in parallel. How does this high throughput capability impact the drug discovery process?
The impact is substantial. High throughput screening, HTS, is a critical step in early drug discovery, where we need to evaluate a large library of compounds to identify those that show activity against a particular biological target, such as an enzyme, receptor, or protein complex. Traditional HTS methods are often expensive and time-consuming, not to mention the large volumes of reagents they require. With microdroplets, we can perform thousands of assays simultaneously in a much smaller footprint. This increased throughput accelerates the screening process and reduces costs significantly. Moreover, because each droplet is isolated, the risk of cross-contamination is minimized, which improves the accuracy and reliability of the screening data.
That isolation sounds particularly beneficial. Can you elaborate on the technical aspects of how these microdroplets are created and controlled within microfluidic systems?
Absolutely. The creation of microdroplets typically involves forcing two immiscible fluids, usually an aqueous solution containing the compounds and targets, and an oil phase, through a microfluidic junction. At this junction, shear forces break the aqueous stream into discrete droplets. The size and frequency of the droplets can be precisely controlled by adjusting flow rates, channel geometries, and the properties of the fluids involved. Once formed, these droplets can be manipulated through the microfluidic network using techniques like electric fields, pressure-driven flows, or even optical methods. Additionally, the droplets can be merged, split, or incubated under controlled conditions, allowing for complex multi-step reactions to be carried out in a highly parallelized fashion.
That level of control is impressive. How does this technology enable more efficient high-throughput screening compared to traditional plate-based assays?
In traditional plate-based assays, each well in a multi-well plate represents a single reaction chamber. These wells are relatively large, and handling hundreds or thousands of plates quickly becomes resource-intensive and laborious. In contrast, microdroplet systems can generate thousands to millions of droplets per minute, each serving as an individual reaction vessel. This not only reduces the time required for screening, but also cuts down on reagent consumption. For example, instead of using milliliters of expensive compounds, we use mere nanoliters per droplet. Furthermore, microdroplet systems allow for rapid mixing and shorter reaction times due to the small scale of the droplets, which translates to faster readouts and quicker decision-making in the screening process.
I imagine that with such a high number of individual assays, data management becomes a significant challenge. How do you handle the vast amounts of data generated during these high throughput screens?
Data management is indeed a critical aspect of microdroplet-based HTS. The sheer volume of data generated requires sophisticated image analysis, fluorescence detection, or other sensor-based readouts, often integrated with automated data acquisition systems. Advanced algorithms, including machine learning techniques, are frequently employed to process and analyze the data in real time. These algorithms can identify active compounds by analyzing parameters like fluorescence intensity, reaction kinetics, or other measurable outputs from each droplet. By integrating these systems with cloud-based platforms, researchers can store, analyze, and share data efficiently, enabling rapid decision-making and iterative screening cycles.
That integration of data analytics with microfluidics seems to be a game changer. Speaking of which, can you provide an example of how microdroplets technology has led to a breakthrough in drug discovery?
Certainly. One illustrative example comes from a recent study on enzyme inhibitors. Researchers were looking for new compounds that could inhibit a particular enzyme involved in a metabolic disorder. Using a microdroplet-based HTS platform, they screened a library of over 10,000 compounds in just a few hours. Each droplet contained the enzyme, a substrate, and one test compound. By monitoring the fluorescence signal indicative of the enzyme's activity, they were able to rapidly identify several candidate compounds that significantly reduced the reaction rate. Subsequent validation and optimization of these compounds led to the development of a promising drug candidate that is now undergoing further preclinical testing. This example showcases how microdroplets technology can accelerate the early phases of drug discovery by enabling rapid, efficient, and cost-effective screening.
That's a powerful demonstration of how the technology works in practice. However, some critics argue that microdroplet systems might oversimplify complex biological interactions. What do you say to those who claim that results from these miniaturized systems might not translate well to in vivo environments?
It's a valid concern. Microdroplet systems by design create highly controlled and simplified environments that might not capture all the complexities of in vivo biology. However, the goal of early-stage drug discovery is to identify promising compounds that show activity against a target in a controlled setting. Once these compounds are identified, they undergo further validation in more complex cellular and animal models. In many cases, the high sensitivity and reproducibility of microdroplet assays can actually help narrow down the pool of candidates more efficiently. Additionally, researchers are continually working on integrating more sophisticated microenvironmental factors into microfluidic systems, such as co-cultures of multiple cell types or gradients of signaling molecules, to better mimic in vivo conditions. So while no in vitro system is perfect, microdroplets provide a valuable, high-throughput platform for initial screening.
That makes sense. The idea is to use microdroplet screening as an early filter before moving to more complex and costly systems. Now, let's delve into the concept of high throughput screening itself. How do microdroplets enhance the efficiency of HTS, and what does target of interest mean in this context?
High-throughput screening refers to the process of rapidly testing a large number of compounds against a specific biological target, which could be an enzyme, receptor, ion channel, or even a cellular pathway. The target of interest is the specific protein or pathway that is implicated in a disease or biological process. Microdroplets enhance HTS efficiency by miniaturizing each individual assay, which allows for thousands or even millions of reactions to occur in parallel. This is achieved by compartmentalizing each reaction within its own droplet, thereby minimizing crosstalk between assays and ensuring high reproducibility. Moreover, the small volume of each droplet means that reactions can proceed faster and with less reagent waste, making the screening process both quicker and more cost-effective. The rapid generation and manipulation of microdroplets mean that researchers can iterate through different conditions and compound libraries far more rapidly than with traditional well plate methods.
The parallelization is truly impressive. With this approach, how do you ensure that the microdroplets are uniform in size and content, given that any variability could affect the screening results?
Uniformity is indeed critical for reliable high-throughput screening. To achieve this, microdroplet generation is typically performed using precisely engineered microfluidic devices that control fluid dynamics with high accuracy. Factors such as channel geometry, fluid viscosity, and flow rates are meticulously calibrated to produce droplets of consistent size. In many systems, the droplets are generated using flow focusing or TJ junction geometries that ensure high uniformity. Additionally, real-time monitoring systems are often incorporated into the microfluidics setup to continuously check for variations in droplet size and composition. If any deviation is detected, the system can automatically adjust the flow parameters to restore uniformity. This level of control is essential because even slight variations can lead to differences in reaction kinetics or detection sensitivity, which would ultimately affect the screening's reliability.
Speaking of detection sensitivity, can you discuss how researchers measure the outcomes of these high throughput assays within each droplet? What kinds of detection methods are typically used?
The detection methods in microdroplet HTS are quite diverse and depend on the specific assay being performed. Fluorescence detection is one of the most common techniques. For example, if an enzyme reaction produces a fluorescent product, high-speed cameras or photodetectors can be used to measure the fluorescence intensity in each droplet. Other methods include absorbance measurements, luminescence, and even mass spectrometry for more detailed chemical analysis. Some advanced systems use integrated sensors that can detect changes in electrical impedance or other physical properties. The choice of detection method is driven by the nature of the biological reaction and the properties of the target molecule. What's remarkable is that many of these detection systems can be integrated directly into the microfluidic platform, allowing for real-time monitoring of reactions as they occur within each droplet.
That integration certainly adds a layer of efficiency to the process. Now, let's explore some challenges. What are some of the current technical hurdles in using microdroplets technology for high throughput drug screening, and how are researchers working to overcome them?
There are a few key challenges that we're addressing. First, while the generation of uniform microdroplets has advanced considerably, maintaining stability over extended periods remains a challenge, especially when reactions require longer incubation times. Researchers are exploring new surfactants and stabilization techniques to prevent droplet coalescence and evaporation. Second, integrating complex multi-step reactions within droplets, such as sequential reagent addition or controlled mixing of multiple droplets, can be technically demanding. This is being tackled by designing more sophisticated microfluidic architectures that allow for precise droplet manipulation, including merging, splitting, and sorting. Third, the vast amount of data generated requires robust data management and analysis tools, as we discussed earlier. Advances in machine learning and real-time data processing are being applied to handle these data streams efficiently. Overall, while challenges remain, continuous innovation in microfluidic engineering, material science, and data analytics is steadily pushing the boundaries of what's possible.
It sounds like a highly interdisciplinary effort. How do you see collaboration between engineers, biologists, chemists, and data scientists driving further innovations in microdroplet-based drug screening?
The collaboration is absolutely crucial. Drug discovery is inherently an interdisciplinary field. Engineers design the microfluidic devices that generate and manipulate the droplets, while biologists and chemists develop the assays and interpret the biological relevance of the results. Data scientists, on the other hand, create algorithms to analyze the massive data sets produced by high-throughput screening. When these disciplines work together, the synergy can lead to breakthroughs that would be impossible in isolation. For instance, an engineer might optimize the droplet generation process, but it's the biologist who will pinpoint how those droplets can best be used to mimic cellular environments or biochemical reactions. Data scientists then take over to refine the screening process by identifying patterns and correlations in the data. This seamless integration of expertise accelerates the drug discovery process and opens up new avenues for innovation.
That's a compelling vision for the future of drug discovery. Shifting focus a bit, let's discuss the impact of microdroplets technology on the discovery of drugs targeting specific diseases. Can you provide an example of a target or disease area where microdroplet screening has been particularly effective?
One notable example is in the search for inhibitors of kinases, a class of enzymes that play key roles in cell signaling and are often implicated in cancer. Kinase inhibitors are a major focus in oncology drug discovery. But screening for these compounds using traditional methods can be challenging due to the complexity of kinase signaling pathways and the need for precise reaction conditions. Microdroplet platforms have enabled researchers to screen large libraries of compounds against specific kinase targets with remarkable speed and efficiency. By encapsulating the kinase reaction in microdroplets, researchers can measure the inhibition effect in real time through fluorescence-based assays. This approach has not only accelerated the identification of potential inhibitors, but has also allowed for the fine-tuning of reaction conditions to better mimic the intracellular environment. The result has been the discovery of several promising drug candidates that are now advancing through preclinical studies.
That's an excellent example. It really highlights the potential of microdroplet technology to speed up the discovery process in critical therapeutic areas. On a related note, how does the use of microdroplets affect the way researchers validate their findings before moving on to more advanced stages of drug development?
The microdroplet approach excels at the early stages of drug discovery by providing rapid, high-resolution screening data. Once a set of promising compounds is identified via microdroplet screening, those compounds typically undergo further validation using more complex assays. This might include cell-based assays, where the compounds are tested in living cells or even animal models. The advantage of starting with microdroplets is that you can quickly eliminate a vast number of compounds that show no activity, focusing resources on a much smaller subset that demonstrates potential. Moreover, because the microdroplet assays are so highly controlled, the initial data are often more reproducible and reliable, which helps in the subsequent validation steps. In essence, microdroplet screening serves as a powerful triage tool that streamlines the drug discovery pipeline by identifying the most promising candidates early on.
That efficiency in filtering candidates is clearly a major advantage. However, I'm curious, are there instances where the microdroplet screening might miss potential hits due to its inherent limitations? What are the risks of false negatives in such systems?
False negatives are always a concern in any screening system. In microdroplet assays, one potential issue is that the extremely small reaction volumes may not perfectly replicate the conditions found in larger scale or in vivo environments, which could, in some cases, lead to the exclusion of compounds that might have worked under different conditions. Additionally, if the detection method isn't sensitive enough or if the reaction kinetics are too slow to be captured within the time frame of the microdroplet assay, there's a risk of missing subtle but important interactions. To mitigate these risks, researchers often use multiple assay formats and complementary detection techniques. They might also adjust the incubation times or perform secondary screens on compounds that were borderline in the initial assay. The goal is to design the system with sufficient redundancy and sensitivity to minimize the likelihood of false negatives while still benefiting from the high throughput and low volume advantages of microdroplet technology.
It seems like careful calibration and multiple layers of screening are essential to ensure reliable outcomes. Now let's delve into another provocative aspect, the potential cost implications. Some critics argue that while microdroplet technology is efficient in theory, the initial setup and specialized equipment can be prohibitively expensive. How do you address concerns about the cost effectiveness of this technology in a competitive drug discovery market?
That's a very pertinent issue. While it's true that the initial setup for microdroplet systems can require a significant investment in terms of specialized microfluidic chips, precision pumps, and advanced detection systems, the long-term cost savings often outweigh these initial expenses. The reduced reagent volumes, faster screening times, and higher throughput mean that over time, the overall cost per assay is much lower compared to traditional methods. Moreover, as the technology matures and becomes more widely adopted, economies of scale tend to bring down the cost of the hardware and consumables. Many academic institutions and biotech startups have already demonstrated that microdroplet-based platforms can be scaled up in a cost-efficient manner. In a competitive market where speed and efficiency are crucial, the long-term return on investment for microdroplet technology can be very attractive.
That's encouraging to hear. Now, I'd like to explore how microdroplets technology might integrate with other emerging technologies. For instance, what role do you see artificial intelligence playing in optimizing microdroplet-based high-throughput screening?
Artificial intelligence is poised to play a transformative role. AI algorithms can process and analyze the vast amounts of data generated by microdroplet assays, identifying subtle patterns and correlations that might be missed by traditional analysis methods. For example, machine learning models can be trained to recognize specific fluorescence signatures that indicate successful drug-targeted interactions. They can also optimize the screening process in real time by adjusting parameters such as droplet size, incubation time, and reagent concentration based on ongoing results. By integrating AI with microdroplet technology, researchers can not only speed up the screening process, but also improve its accuracy and predictive power. This integration allows for a more adaptive and intelligent system that continuously learns and optimizes itself, ultimately leading to a more efficient drug discovery pipeline.
That kind of synergy between technology and biology is really the future of drug discovery. Before we conclude, could you share your vision for how microdroplet technology might evolve in the next decade? What breakthroughs or improvements do you expect to see?
I envision several exciting developments. First, I expect the integration of microdroplet platforms with even more advanced detection and analysis techniques, such as real-time mass spectrometry and high-resolution imaging, will push the boundaries of what we can measure within each droplet. Second, the ongoing miniaturization and parallelization of these systems will lead to the creation of fully automated lab-on-a-chip platforms that can screen millions of compounds in a single run with minimal human intervention. Third, as we incorporate more sophisticated AI and machine learning tools, the systems will become increasingly self-optimizing, reducing the need for manual calibration and significantly speeding up the discovery process. Finally, I believe we'll see microdroplet technology being applied beyond traditional drug discovery, such as in personalized medicine, where patient-specific compounds can be screened rapidly, or in the discovery of compounds that address emerging global health threats. Overall, the field is moving toward a future where high-throughput screening is not just faster and more efficient, but also smarter and more adaptable.
That's a compelling vision for the future of drug discovery. As we wrap up, what would you say is the key takeaway for researchers and companies considering adopting microdroplet technology in their own drug discovery efforts?
The key takeaway is that microdroplet technology offers a unique combination of speed, efficiency, and precision that can significantly accelerate the early stages of drug discovery. By miniaturizing reactions and enabling massively parallel screening, it allows researchers to sift through vast libraries of compounds quickly and cost-effectively. While there are technical challenges and initial costs to overcome, the benefits in terms of reduced reagent consumption, improved reproducibility, and the potential for integration with advanced data analytics make it a highly attractive option. For companies and researchers, adopting this technology could mean a competitive edge in bringing innovative therapies to market more rapidly and with greater confidence in the screening results.
Dr. Kim, your insights have been incredibly valuable today. Before we close, could you share a final thought on how microdroplet technology is shaping the future of drug discovery and what you hope to see in the coming years?
I'd say that microdroplet technology is a perfect example of how interdisciplinary innovation can transform traditional processes. It combines principles from fluid dynamics, microengineering, and data science to address one of the most critical challenges in modern medicine, finding effective drugs quickly and efficiently. I hope that In the coming years, we'll see this technology become a standard tool in drug discovery, not just in academic labs, but across the pharmaceutical industry. Ultimately, the faster we can identify promising drug candidates, the sooner we can develop therapies that save lives and improve health outcomes. It's an exciting time to be working in this field, and I'm optimistic about the breakthroughs we're on the verge of achieving.
Thank you, Dr. Kim, for sharing your expertise and providing such a detailed look at how microdroplet technology is revolutionizing drug discovery. Your explanations, from the basics to the advanced integrations with AI, have given us a comprehensive understanding of the subject.
And thank you to all our listeners for joining us on this in-depth exploration of microdroplets and high-throughput screening in drug discovery. Remember, every tiny droplet holds the potential for a breakthrough that could change lives. Stay curious, keep innovating, and continue pushing the boundaries of science. Thank you for listening, and may your pursuit of new discoveries be as relentless as the droplets in our microfluidic systems.
Goodbye, and until next time, keep exploring the world of innovation in drug discovery.